All right, so now we're going to cover chapter 4. We're going to look at the functional anatomy of prokaryotic and eukaryotic cells. We're actually going to focus mostly on prokaryotic cells in this chapter. So guys, if we look here at prokaryotic versus eukaryotic cells, this is a little bit of a review back from chapter 1. So with prokaryotic cells, these are going to be cells that come from the group bacteria, and we're going to focus on bacteria only, not archaea, since archaea does not have any pathogenic members. One thing to remember about prokaryotes is that they do not have a membrane-bound nucleus where DNA is found. The DNA is going to be found in an area that's called the nucleoid, and it's not bound by any kind of membrane. Also, the DNA is going to be in a single chromosome. This chromosome is in a circular loop. Also with prokaryotes, when we look at the organelles, they don't have any membrane-bound organelles. Now they do have ribosomes, but this ribosome structure is a little different than the ribosome structure that we see in eukaryotes. In eukaryotes, this is going to be the groups with protista, fungi, plantae, and animalia. They do have a membrane-bound nucleus that is going to contain the DNA. The DNA is found in chromosomes, which means there are normally plural, more than one chromosome, and they are associated with histone proteins. Eukaryotes also have various membrane-bound organelles. These include things like mitochondria, rough ER, smooth ER, Golgi apparatus, those sort of things. They do still have ribosomes as well to help with protein synthesis, but their ribosome structure is a little more complex. All right, when we look at the ribosomes, prokaryotes have a 70S type ribosome and eukaryotes have an 80S. All right, so these are some of the differences between prokaryotic cells and eukaryotic cells. When we also look at prokaryotes, when we look at bacteria, typically contain a cell wall that contains peptidoglycan. That is one of the main components that are part of that peptidoglycan, which the peptidoglycan is one of the main components as part of their cell wall. And you can see that here in this picture. When we talk about the eukaryotes, in some of them, there's no cell wall present. But if it is, it's normally simpler than what we see with the prokaryotes. When we look at fungus do have a cell wall made out of chitin or chitin. Plants have a cell wall made out of cellulose, which is a type of sugar, and Animalia does not have a cell wall. And this means it's absent. All cells have a plasma membrane. However, not all cells have a cell wall. So all cells do have to have the plasma membrane present, but they do not have to have a protective cell wall. And if you look here, this also shows you the picture of a fungal cell wall. Continuing on with the prokaryotes versus eukaryotes, if we look at cell division, when we look at bacteria, they are going to undergo what we call binary fission. It is a type of asexual reproduction, meaning that it comes from one parent. The cell is going to split into two. One thing to note here, though, is this is not mitosis because there's no nucleus. Mitosis means the splitting of a nucleus, and prokaryotes do not have that. Okay, so that's why it's called binary fission. On the other hand, with eukaryotes, because there is a nucleus present, this is still a form of asexual reproduction. One cell is splitting into two. However, we are also splitting the nucleus in this process. So since it's asexual reproduction, you need to recall that this is going to create genetically identical parent cells. Also, when we look at binary fission, you need to remember this is because there is no nucleus, and mitosis does involve the division of the nucleus. Now, when we look at cell organization in prokaryotes, they're normally less complex because they don't have all those organelles and those sort of structures. They also are normally unicellular and they're pretty small in size. When we look at eukaryotes, though, they're more complex due to the amount of organelles they do contain. Most of the organisms in the eukaryote group are going to be multicellular. There are some exceptions when we look at the group protista, and they are normally about 10 times larger in their cell size compared to prokaryotes. Okay, so these cells are going to be larger and more complex. All right, so we're going to focus on the prokaryotes here. We're going to be looking at the domain archaea and bacteria. Remember, though, in archaea, none of them cause disease. One thing we're looking at with prokaryotes is we can ID them by their cell shape and their arrangement. So when we're looking at shapes, they could have round shapes, rod shapes, or helical shapes. So when we're looking at this, when you see helical, this is kind of like a spiral. This is why they could be called spirillum, a spirochete, or even what we call a vibrio. And vibrio is kind of a comma-shaped structure. The round shapes are going to be be the coccus, so they are going to have that structure, and cocci is going to be for the plural. The rod shapes are going to be known as bacillus or bacilli, and then of course the helical could be spirillum, spirochete, or the vibrio, which is the comma shaped. All right, so this is what we're looking at for the shapes of the bacteria. When we look at arrangement can be a little bit different depending on what kind of groups we're looking at. This particular one is looking at the arrangement of cocci, the coccus, the round ones. All right, so we can have what we call the diplo, and the diplo means they're in pairs. So two of them are together in a pair. We can also have the strepto, which means they're in chains. This is going to make them look kind of like a string of pearls. We also can have them in forms of packets. This is where they're going to come in like fours, where they're like a tetrad, or they might have kind of a cube shape where there's like octets or 8 of them. And so this is going to be what we would consider more of a packet type of structure. And then we have staphylo, and staphylo is going to be a grape-like cluster. Okay, so it's clustered together like grapes. Now, something that's important to note is that when organisms are grown on artificial medium, like in a lab, we see that they don't normally exhibit their typical cellular arrangement. They will exhibit their typical cellular shape, but not necessarily their arrangement based on the fact that they are growing on an artificial media. So when we see here, this example is for Streptococcus, and for Streptococcus, it should be in a chain-like structure. And we see in the one side that they aren't in chains, but the other they are. And the same thing here with the Staphylococcus. They should be clusters and clusters of like grapes. And again, when it's on the artificial media, it does not show up that way. Okay, so when we look here, these two are going to be grown on the agar plates with the artificial media. Therefore, they don't display the normal cellular arrangement. All right, so in this set of notes, we have like a little area for you to quiz yourself. It's describe the following by Gram reaction, morphology, and arrangement, because we should have already talked about Gram reaction. That was from the previous chapter. All right, so when we look at this first group, this is a Gram positive because it is purple in color. And we would also say this is a diplococci because they are in pairs and they have a circular shape to them. We also see in this one, there's the diplococci because it has them two and they're in that circular shape. However, this one, because it's in a reddish pink color, is Gram negative. This last group is Gram positive. The shape is the cocci, which is round, and they are located in chains. So when we're looking at this can help us give some sort of idea of what we are technically looking at. So it's important, though, to note that just based on the arrangement, just based on the morphology, just based on the Gram stain, we can actually not put these into a particular group of knowing this is the actual bacteria. There are further tests that have to be done. Now, can it lead us towards a certain type? Yes, it can. Whether it's Gram positive or Gram negative, if it's cocci or bacillus, but it can't give us a definitive idea of what bacteria it is. And so on this, we could say this first one could be Streptococcus pneumoniae, but again, we'd have to do some further tests to find out for sure. The next one could be Neisseria gonorrhoeae, and this one could be a form of gonorrhea, and the last one could potentially be Enterococcus. But again, there's question marks here because we'd have to do further tests to find out exactly what type of bacteria we're looking at. Now, when we get to the bacillus group with the rod shape are a little different in what we look at for their arrangements. They could have a single bacillus. We could also look at it in terms like diplobacilli or streptobacilli, but these typically are not used to describe cellular arrangement when we're looking at the bacilli groups, but it is still kind of in chains. We also have the group that's the coccobacillus, where it's almost rod-shaped, but it also is kind of spherical. So this one sometimes will look like that diplococci, but this is what we would call coccolobacillus. It's between those two shapes. Now, the term bacillus is used to describe cellular morphology, the rod shape, which is not the same as the bacillus species when it is capitalized. All right, so when it's capitalized and it's italicized, that's when we're looking at a certain bacillus species of bacteria versus just the morphology or what the cell looks like. So again, we can describe the following based on their Gram reaction, their morphology, and their arrangement. And so if we look here, this is Gram positive bacilli. They have more of like a boxcar type of shape to them. We also have the gram-positive bacilli, but these are also going to be more in chains. So technically you could say it would be streptobacilli, but again, that term's not normally used. When we look at these, you can see in the picture that they all resemble the same type of shape. Therefore, they're known as monomorphic. Mono meaning one, morph meaning shape or structure. So they are going to be what we consider monomorphic. If you look at this other picture over here, though, these are gram-positive and they are bacillus, but if you'll notice, this bacillus has a lot of differences in its shape. Okay, this is what we would call pleomorphic, this is what we would call pelomorphic, meaning that there are multiple shapes to this. Okay, many shapes. These a lot of times get described as looking like Chinese letters or types of clubs in a kind of a palisade arrangement. And palisade just means that it is in an even row of cells. Okay, so when we're looking at palisade arrangements, like an even row of cells. These a lot of times are your diphtheroids. Diphtheroids are going to be the different types of bacteria that cause diphtheria. One of the main groups is the Corynebacterium. This is going to be part of that group. This one is not going to have that distinctive bacillus shape that you see in the monomorphic. All right, so now we're going to move on to looking at structures on the external part of the cell. Now, these structures we're really going to look at are going to normally help with virulence, meaning it's going to cause the bacteria to be more pathogenic, giving it a type of advantage. The first one is the glycocalyx. This is normally a capsule. Again, it is a virulence factor. This is firmly attached to the cell wall of the bacteria. And so when we look here, it's going to be in kind of this goldish color. We also see that some of this, in some bacteria, this structure is more of a slime layer. This is going to be more loosely attached to the cell wall, but both are going to give some sort of advantage to this bacteria. Now, if we look here at this bacterium, the inner layer, that's the yellowish, the light yellow, is the plasma membrane. The middle layer is the cell wall, and the outer layer is the capsule that we're talking about here, what's going to be attached to that cell wall. Now, sometimes this is a gelatinous type of sugar coat. This protects the cell from being engulfed. A lot of times when a bacteria gains access into like an organism like ourselves, our immune system wants to engulf it, take it in, and destroy it. This particular structure prevents that from happening, so it offers the cell some protection against your immune system. We also see that they can have what we call a K antigen. This is the capsular antigen, and this does induce your immune system response, causing your immune system to respond to this invader because it is a different marker that your body does not have, or the organism does not have. Now, going back to our chapter that we talked about staining, we talked about how there are some certain types of stains that we can use to actually visualize these capsules. What type of staining was used to see the capsules? This is that negative staining. The negative staining allows you to see the capsule around the bacteria. So one way to kind of visualize this kind of slime layer that is on some of these bacteria, you can see it when we grow it on this artificial media. It gives it a colony morphology that looks like a mucus. It's like a mucoid looking structure. Some examples of this is the Klebsiella pneumoniae shown here on the MacConkey's agar, and then also Streptococcus pneumoniae, which is on blood sheep agar here in this example. Now, some virulent strands of Bacillus Anthracus is also going to produce capsules in this case. The next structure we want to look at here is the flagella. Flagella is going to help with motility or movement. This is associated with an H antigen. All right, so it's different than what we saw with the capsule or the slime layer, but this one is going to be looking at the H antigen. An example of this is a subtype or a group of E. coli. This is called the E. coli cerevar or O157H7, and the H is what we're talking about here with this particular antigen. This is the protein in the flagella that's useful in distinguishing among different variations of species, like in this case, variations of E. coli. Now, these flagella can be found in different areas on the cell. There could be multiple flagella like you see in some of these pictures, or there could be a single flagella. All right, so when we're looking at this first group, this has many flagella all over it. Okay, this is the trichosis. We then see the monotrichosis, and this one's going to have one. Mono means one, and it's normally polar, meaning it's on one end. Down here in C, you can see it's the elliphotrichosis, or polar. So it has many flagella, but they're all still located on one end of the cell. And then the last one is going to be the amphitrichosis, and they're also polar, but they're found on both poles of the cell. A lot of times with this last one, when we look at D, it's going to have more of a kind of rotational type of movement, kind of like a corkscrew movement. We also see when this happens, this can be what these axial filaments can be for the motility, and you see this in the spirochetes. Okay, the ones that have the real spiral shaped to them. This again is going to allow them to move in a corkscrew type movement with a type of rotation. One of the best examples of this is Tryponium pallidum. This is the causative agent of syphilis. This is also going to be known as the endoflagella. This is fibrils that we're going to wrap around the cell and lays within the outer sheath. Again, this is what helps produce that corkscrew type of movement. This corkscrew type movement allows it to move through fluids very easily. The next structure we want to look at is the fimbriae. The fimbriae is going to help with adhesion, holding on to surfaces or to cells. And also the fimbriae can also extend to make a sex pilus. A sex pilus is to transfer genetic information from one bacteria to another. Since bacteria undergoes asexual reproduction, they all would be genetically the same. However, bacteria is really good at picking up extra pieces of DNA and then hopefully utilizing them for their own benefit. This will allow them then to transfer to them to other individuals, okay, and they do this through the sex pilus. When we look at the fimbriae, they can have these bristle-like fibers, and they could have 200 to 400 of these per cell. This allows the cell to attach to a surface kind of like Velcro, okay, being able to hold on to a surface. If present, it's normally associated with gram-negative organisms, and again, some of these examples are the E. coli, Neisseria gonorrhea, and that sort of thing. The sex pilus is, though, a tubular transfer. They could have none to less than 10 per cell found on a bacteria, and this is used for that bacterial conjugation, where the two sex piluses will merge together, creating a tunnel, and they will be able to share pieces of their DNA. Not their whole DNA, but at least pieces of it. All right, so moving on in, we looked at the outer structures that are connected to the cell wall. Let's look at the cell wall itself. The cell wall is there to protect the plasma membrane, and it also offers the cell shape for that bacteria. If we have a gram-positive bacteria, remember and recall from the last chapter that this is going to have a thick layer of peptidoglycan. This thicker layer is going to allow it to hold on to the crystal violet stain, obviously giving them that purple color for a gram-positive reaction. Now, if you'll notice here in this picture, this peptidoglycan is in kind of a lattice type structure. This is going to be between disaccharides, which are sugars, as well as polypeptides, which are the proteins. In a gram-negative bacteria, this is going to have a thinner layer of peptidoglycan. This thinner layer of peptidoglycan is going to have an outer layer of a lipopolysaccharide. This lipopolysaccharide a lot of times is lipid A. This is an endotoxin that is released when the cell dies, and so a lot of these will have something that when the cell is killed by the immune system or it dies, it releases this toxin that can still potentially cause harm to to the host that it invaded. This O polysaccharide is a somatic antigen. This one is found again on that E. coli serovar that we saw, and that's where the O157 comes from. Okay, so not only does this E. coli have that particular type of flagella we saw with the H, it also has a particular type of polysaccharide that we see here with the O. All right, so this allows us to be able again to distinguish subtypes of different types of bacteria. All right, so if we take a look at the picture here, you can see the layer of the peptidoglycan. You can see that there is a thicker layer though of the lipopolysaccharide. All right, so when we're looking at this, we want to look at a few other things. We see here that we have the mycoplasma. This is a causative agent of walking pneumonia. This group is unique because it lacks a cell wall. It's the smallest living microbe that we know of, and it has a very high lipid content. So it has a lot of sterols in its plasma membrane. We also see Mycobacterium has a very high lipid content. This is the mycolic acid, and this is why sometimes instead of using a gram stain, we would use that acid fast fast stain, okay, because of their unique cell wall. Now, archaea cell walls are different than bacteria, therefore we would have to look at them and study them chemically. We would have to do some different types of tests. Now, one reason we want to know what kind of cell wall is present, whether it's gram-positive, gram-negative, whether they lack a cell wall or their cell wall has lots of fats or lipids to it, the reason we want to know that is because some antibiotics will actually interfere with how they make or synthesize their cell wall. If we're able to interfere with that, it does give us an advantage because now they don't have that wall of protection protecting the cell. All right, penicillin is one of these, and it's very effective against gram-positive bacteria. Therefore, it's going to mess up those cross bridges we saw in that thick layer of peptidoglycan. So guys, bacterial cell walls with atypical cell walls do not gram stain real well. So when we see some of these, this is why we use things like the acid fast and some different other techniques, because they don't gram stain that well. Different antibiotics are used on gram positive versus gram negative, and this is due to the structural differences of their cell wall. And damage of the cell wall could lead to osmotic rupture of the plasma membrane. One thing to note is animal cells like our cells, if our cells get put into a solution that's hypotonic, the cell will ultimately start to swell and it would burst. When a cell has a cell wall around it, though, it helps protect them against this, okay, keeping them from potentially being affected by that fluid movement as much. If we can put holes into the cell wall or cause it not to be formed properly, it makes it more susceptible to this rupture of the plasma membrane. So guys, here we have table 4.1. It's looking at gram-positive versus gram-negative bacteria, and we're going to do some comparisons. All right, so gram-positive retains the crystal violet, so it has a purple color. The gram-negative is going to take in the counter stain, which is the saffron, giving it the reddish pink color. If we look at the peptidoglycan layer, gram-positive has a thick layer, gram-negative has a thin layer. If we look at periplasmic space, or the outer lipopolysaccharide or endotoxins released, gram-positives don't have this because they don't have these structures present. But in a gram-negative, they are present, and one of the main lipids that are present that acts as the endotoxin is lipid A. When we look at sensitivity to things like lysozymes or penicillin, gram-positive are very highly sensitive to these. So these are a good choice in order to take out gram-positive types of bacteria. However, for gram-negative, they are not as useful. All right, they have a low sensitivity to this. And then resistance to drying out, gram-positive, since they have a very thick layer of peptidoglycan, they don't dry out as easily. When the environment gets dry, they can resist that change for a longer period of time. Whereas gram-negative, due to the thin layer, they are not able to do this. All right, and so when we look at this, if they have a high ability to withstand drying, you're going to find them they're going to be very prevalent in the environment. Okay, so they can be in soil, things like that. All right, so moving in deeper, we are going to get to the plasma membrane. The plasma membrane is also known as the cell membrane or the cytoplasmic membrane. The structure here of this membrane is what we would call a fluid mosaic model. Now, this means it has kind of a fluid structure due to its phospholipids. It's kind of like a sea of fats, but inside of the sea of fats, there are proteins embedded, and you can see them here in like this purplish blue color. These proteins are going to help and be floating kind of like buoys in this ocean of fats, and this is why it's the fluid mosaic model. Now, going back to this structure, when we look here at the phospholipids, it does form a phospholipid bilayer, meaning there's two layers of these phospholipids. These phospholipids have a hydrophilic head region that's shown in red. These are polar and they actually like water, whereas the yellow region on the inside is going to be the hydrophobic tails, which means they don't like water, and this is what creates that selective barrier for the cell. We also see in here with the mosaic part of the structure, things that are embedded into the sea of fats are the integral proteins. They are going to run completely across the membrane. We also see peripheral proteins, and peripheral proteins are going to be normally on one side. Okay, think about peripheral vision, which is what you see out here on the sides. Peripheral proteins are only going to be located on one side of the membrane. Some other structures that we see located here in this particular membrane are things like glycolipids, cholesterol, glycoproteins, and carbohydrates. However, in a lot of bacteria plasma membranes, they actually lack the glycoproteins, the glycolipids, and the sterols, okay, as well as the carbohydrates. They don't have these markers like we see on a lot of the other plasma membranes. There is an exception though. The mycoplasms do contain these. So the function of this plasma membrane, of course, is to act as a barrier. It's a barrier between the inside of the cell and the outside of the cell, so the external environment. It is subject to rupture, so it doesn't really have a protective type of structure here. That's what the cell wall is for, but it does help the cell transport substances. Whether it's going to bring things into the cell or send things out of the cell, this is going to be the area that is going to be responsible for that movement or transport. The plasma membrane is considered selectively permeable. It's going to be very selective on what gets to move in and what gets to move out. You can kind of think about it as like a bouncer of like a high-end club. They're going to get to pick who gets to come in, and if you are being disruptive, then they will then kick you out. All right, so that movement of who gets to come in and out of that club is determined by that bouncer. In this case, the bouncer is the plasma membrane. It's determining what gets to come in and what gets to leave the cell. So just for your information, guys, when we look at the bacteria plasma membrane, this is where aerobic cellular respiration is going to occur. We're going to talk about aerobic cellular respiration in Unit 2, but cellular respiration normally, like in our cells, takes place in the mitochondria, but since these guys don't have organelles, they don't contain mitochondria, this is going to be the structure. The plasma membrane is the structure going to be responsible for cellular respiration. I'll recall that cellular respiration is going to be how the bacteria is going to get ATP or energy. All right, so we talked about this plasma membrane being an area for transport or movement of molecules across this barrier. There's different ways that we can actually move these molecules, and so some of them are through what we call active transport. Now, active transport means that the cell actually has to do some work. Since it has to do work, it's going to require ATP. ATP is the energy source. This is going to make things move in a way that they don't want to go. So it's going to pump molecules across their gradient. Now, if you'll recall back from biology, you learned that molecules like to move from high concentration to low concentration. They want to move that way until things are even. All right. In the case of active transport, we want to have, if it's high over here, we want to push more over here. This means it's going to require energy to do this. All right. If you've ever seen, like, if you have a toddler, or if you've ever dealt with a toddler or seen like a parent deal with a toddler at a store, and they don't want to go somewhere, and then they start to throw a fit and they fall down and they're like dead weight, and the parent's trying to physically move them and take them where they want them to go. That's kind of how these chemicals are working. They're working against the cell, but the cell is using energy to push them where they don't want to go. On the other hand, there's passive transport. Passive transport has no energy is required from them. So when we're looking at passive transport, this is that whole idea of the molecules being from high concentration and they're moving on their own to low concentration. And the cell just utilizes this, all right, moving things from high to low. This would be like getting into the lazy river. Once you get into the lazy river and you get into your tube, it just takes you around. The current of the lazy river does the work for you. You don't actually have to do anything, all right? And so that's what we're looking here at the passive transport. The cell doesn't have to do anything. The molecules are doing the work. They are moving from high concentration to low concentration. All right, so when we take a look at these types of transport, if we're looking at passive transport, they could be just through simple diffusion. Like you see here, these molecules are simply diffusing through the plasma membrane from high to low. They don't need any help doing this, and no ATP is required. In some of these molecules, they have a hard time crossing the membrane itself due to that fat structure, so they just need some help to get across. Still, no energy is required, but they may need a protein, and these integral proteins that we saw that went all the way through the membrane can act as channels or carriers for these molecules. The last one over here is osmosis, and osmosis is the passive transport of specifically water. All right, so osmosis is where we're going to see water going from high concentration to low concentration. All right, since we're talking about osmosis, let's kind of stay on this for a little bit and see what happens to cells based on this water movement. So osmosis is the movement of water from high concentration to low concentration across the selectively permeable membrane. So in this case, the plasma membrane. This brings us to a term called tonicity. Tonicity of a solution describes how much stuff is dissolved in it. Okay, the solute concentration. So is there salt dissolved? Is there calcium dissolved? There's things dissolved in it. And we represent this here with just this little circle, this grayish circle. Okay, so when a solution is what we would call hypertonic, okay, it means it has more solute and less water. So in this picture, you can see that there's more of those little dots on the outside of the cell than on the inside. All right, so the outside is what we would call hypertonic. All right, it has more solute, less water. A hypotonic solution is going to have less solutes and more water. So if you look at the picture here, you'll notice that there's only one solute outside of the cell and there's more inside. Okay, so this is what we would call a hypotonic solution. We can also have an isotonic solution, and iso means equal or same. So an isotonic solution is where there's an equal amount inside and outside of the cell. All right, so this is where they've actually come to an equilibrium where it's an equal amount. So one thing to note though is water is always moving. It's always moving through osmosis. And when we look at this, water is always going to move from its high concentration to its low. And so because of this, it's always going to move from Po to Per. It's going to move from a hypotonic area to a hypertonic area, because this is where we're looking at water, not the actual solutes. Okay, so it's the movement of water. So cells are going to react a little different based on what kind of solution they are put in. So in this case, if a cell is put in a hypertonic solution, the cell is considered hypotonic and the water will leave the cell. This will cause the cell to start to shrivel up because it's losing water. On the other hand, if the cell is put in a hypotonic solution, like you see in the second picture, then water is going to enter the cell, causing the cell to start to expand. Now, in the case of bacteria, as long as they have a cell wall, this will help keep the cell from potentially rupturing, okay, like a balloon, like when it gets too much air or water in it and burst. But if the cell wall is not there, it could burst the cell. When we look at an isotonic solution, water is still going to move, but it's going to move equally. If 5 water come in, 5 are going to go out. If 10 water come in, 10 are going to go out. It's an equal type of movement. So nothing happens to the cell. Okay, the cell doesn't change shape or size in any way. So let's just review this real quick. If a cell is placed in an isotonic solution, okay, there's going to be no net movement, meaning no gain or loss of water. Water will still move, but it'll be equal, so nothing happens to the cell. If a cell is placed in a hypotonic solution, the cell will start to swell. Okay, it will take water in because remember it moves from Po to Per. This is going to cause osmotic lysis to occur, and this is going to happen if the cell wall is damaged. Well, there are a number of ways we could damage the cell wall of an invader, of a bacteria that's making us sick, and we could do this through enzymes like lysozyme activity or antibiotics, medications. On the other hand, if the cell is placed in a hypertonic solution, the water will leave the cell, causing the cell to shrink. This is called plasmolysis. All right, the plasma membrane starts to pull away from the cell wall because it's shriveling up and shrinking due to the loss of water. Now, the reason we want to talk about this movement of water is this actually can help us control microbial growth. Okay, if we're looking at growing microbes in a culture on a plate, we need to make sure that there's a perfect osmotic condition. This way the bacteria can grow in or on the media properly, so the cells aren't bursting or shriveling up and shrinking. Bacterial cells in a hypotonic and hypertonic solutions are subject to becoming metabolically inactive, and if they don't have an active metabolism, then they're not going to actively be dividing and creating more cells. Therefore, it's going to be difficult for us to grow it. Okay, so it's important for us to, when we want to grow these things in the lab on media or in broth, we want to make sure that the conditions are going to be in like more of that isotonic type of solution so that the cells can flourish and they can start to grow. So some other structures we want to look at in a prokaryotic cell are going to be the cytoplasm. The cytoplasm is composed mostly of water. However, it also has a variety of inorganic or organic substances located in it. This is an aqueous solution that's going to bathe the nucleoid or DNA region and the ribosomes. So this is what the nucleoid or DNA is floating in, as well as the ribosomes inside of the cell. The next structure we see here is the nucleoid. The nucleoid is the region or area that the DNA is located. Remember, since this is a prokaryotic cell, the DNA is a single loop. However, it is still double-stranded because DNA is double-stranded, but it is a single loop of DNA. Okay, so they have one chromosome. Now, if you'll notice in here, it doesn't look like it's just one, but it is one single loop that's wadded up. Okay, think about it in the sense of like you have a bunch of yarn or string and you tie the two ends together. It's a single loop, but then it could get kind of wound up and tightened up in that process of fitting into a small space. There's also some things called plasmids in here. Plasmids are accessory rings of DNA. They're small rings of DNA that are not located in that central main chromosome. These are not crucial for metabolism. These are going to be extra genes. These extra genes are going to be present in order to help the bacteria be able to survive. These are the things that are going to get passed through the sex pili we talked about when they come in contact and they share and it creates that bridge or tunnel. These are the pieces of DNA that will move across that tunnel. These can be responsible for antibiotic resistance. When we find that bacteria starts to become antibiotic resistant against the antibiotics that we use, this is normally the reason why. They have this extra set of genes that helps protect them and they're sharing it with all of their friends. We also see that these can also produce toxins. They can produce toxins that can help the bacteria again survive as well. We use these in biotechnology to do gene manipulation. We can actually inject some plasmids into bacterial cells for them to make insulin for us or growth hormone or things like that that the bacteria doesn't need but we as humans need, and it makes the bacteria make them and we package them and sell them at the pharmacy, but we do utilize this in gene manipulation tactics. All right, so this brings us to the ribosomes. Now, ribosomes are found both in prokaryotic cells and eukaryotic cells, but their structures are a little bit different. Okay, ribosomes, if you'll recall, are responsible for protein synthesis. This is going to be where translation takes place, where we're going to translate the information from the DNA and RNA, and we're going to translate it into a protein. Now, ribosomes are made of two units or subunits. There's a small unit and a large unit. One reason why this whole idea of the ribosomes being different is important is some of the antibiotics we use target protein synthesis, and it means they target the ribosomes. So if our ribosomes were the same as the bacteria's ribosomes, that means the antibiotic would also hurt our cells. So the fact that these ribosomes are different in their structure allows us to use medications with certain antibiotics to target these. Now, when you're looking at the S here, guys, the S is what we call the Svedberg units. This measures the sedimentation rate of these different units. So if we look here, the small subunit is a 30S in prokaryotes. The large unit is a 50S. When we put these together, it's a 70S ribosome. Now you're thinking, wait a minute, 50 plus 30 is not 70, it's actually 80. There is a little bit of a difference in this. When they are added together, it changes a little bit of their sedimentation. We don't just add the 50 to the 30. You're going to see the same issue with the eukaryotic ribosomes. Now in the eukaryotic ribosomes, the small unit is a 40S and the large unit is a 60S. When these merge together, they create an 80S ribosome. Okay, so again, 60 plus 40 should equal 100, but when we see the sedimentation rate, when it's combined, it's more of an 80S unit. These 80S ribosomes are not affected by antibiotics. The smaller 70S are. So this allows that medication to be selective and it will only fight against that bacteria instead of fighting against the potential host cells, so your cells. Another thing we want to look at are what we call endospores. Endospores are metabolically inactive structures of a bacteria. This is where the bacteria kind of goes into a dormant or sleeping stage. This is where it dehydrates the cell with thickened layers of peptidoglycan. The purpose for this structure is for survival when conditions are not favorable. Now, these cells can't reproduce at this time, but they can survive. All right, and when conditions improve and they get better, then they could potentially come out of this sleeping state and they can produce more of the bacteria. These endospores can be very hardy and they can actually survive in the environment for years. All right, and so they can survive in the environment for years if they can make this endospore type of structure. Now, once they break out of this and they start to wake up in a sense, because since this is a dormant stage, they kind of start to wake up. That's the germination. They start to grow again. These cells can return to what we call the vegetative state, and this is where they become metabolically active and they can start reproducing at this point. All right. They're only going to do this though if conditions start to improve. Okay. And they have a chance for survival and reproduction, not just survival. Now, a concept here is these guys must be killed during a sterilization process because they're able to go into that dormant type stage and they're able to build up a wall around themselves. This makes it harder for us to potentially treat them with antibiotics, things like that, especially if they're on a surface, because they can be on that surface for a long time. And so one way to help get rid of these is through a sterilization process, which we'll talk a little bit more about in other chapters. So some examples of gram-positive endospore producing bacilli are things like Clostridium. Clostridium is a group that can cause things like botulism, tetanus, or gas gangrene. So when we look at botulism, this is going to release a toxin that will actually cause the muscles to be paralyzed. This is going to be the toxin that they use in Botox. Tetanus, on the other hand, is where it produces a toxin that causes the muscles to contract, causing cramps. So it's the opposite of botulism. And then gas gangrene is where it starts to eat away at the tissues, causing the tissues to die, and it produces a nasty gas in the process. Another example are the bacillus species. Some of these cause food poisoning for the individual, and this group also contains the anthrax. And you may have heard of anthrax before whenever it had been mailed to the Pentagon, and when individuals would breathe in those endospores, it would give them a chance then to have a better environment and they'd start growing, and it could kill the individual very quickly. The implications of this is that we have proper food preservation. So the canning process, when we want to can foods so that we can have them for a longer period of time, we need to make sure that they're done correctly. If they're not done correctly, it allows these endospores to survive in these structures so that when you open it up and you eat it, it can cause the food poisoning or it can cause botulism, things like that. We also see the importance of vaccines in this as well as public awareness. A lot of times with public awareness, we need to let people know that if you step on a rusty nail, that is potentially going to allow endospores that cause tetanus to get into your body, and it gives them a good environment and they want to start to grow. Well, one way to help with that is to get the tetanus shot to help protect you against that. All right. And so when we look at this, public awareness is really important on this. This is one reason too, when they have recalls on things, or if they find that there has been a contamination in a canned food product or something that's causing food poisoning, they will want to pull it off the market and also get you to throw it away. That public awareness is there to help keep you safe. So guys, when we talk about eukaryotes and their structures, we're going to actually do that mostly in chapter 12, which is what we're going to look at next. These examples are Protista. We're going to focus mostly on the protozoa. However, we'll look a little bit at algae. The fungi, we're going to focus on the yeast and the molds. And in the group Animalia, we're going to focus in on the parasitic worms. So these are the groups we're going to focus in on based on the microbiology. All right, but their structures and stuff are going to be similar based on what group they are part of. Now, some of these have flagella, the tails that help them move. Others have cilia, which are hair-like extensions. And then some have cell walls, whereas others don't. These guys, however, since they are eukaryotes, do contain membrane-bound organelles as well as a nucleus. Now, you do need to read about these in pages 94 through 102 in your textbook, but make sure that you also pay attention to the next lecture when we get to chapter 12.